The overall goal of this procedure is to identify afferent neurons by retrograde dye labeling and to study the ion channels in these neurons using patch clamp electrophysiology and immunocytochemistry. This is accomplished by first injecting dye into the target tissue to label the innervating afferent neurons. The second step is to isolate afferent neurons from dorsal root ganglia. Next, the labeled neurons are patch clamped to record the voltage-gated ion channels. The final step is to identify expressed ion channels in the labeled neurons using immunocytochemistry. Ultimately, using a combination of patch clamp and immunocytochemistry, we are able to identify the expression pattern as well as the functional properties of the ion channels in afferent neurons.
The main advantage of this method over other existing method is that it's a non-invasive method, and the dye that we use, dye I, is known for its rapid uptake by the neuronal terminals, lack of transfer between neurons, and long-term retentions within the neurons that we inject. Demonstrating the procedure will be myself and Stephanie McGrew, research technician from our lab.
For dye injection, first shave the hair over the calf area on both legs of the anesthetized rat using an electric razor. Next, fill a 1 cc sterile syringe with 200 microliters of 1.5% dye solution. Slowly insert the needle parallel to the long axis of